everyone, me and my teammates will be explaining about the p53 transcriptional gene network. I will be explaining about the introduction, Yip Teng will be explaining about the key steps, Becky will be explaining about the consequences, and Adeline will be explaining about the solution. So let's start. p53 is a protein known to play its role in the transcription process and acts as a tumor suppressor. This protein is in charge of cell division and acts as a stopping signal. This protein will also work with other proteins to carry out its other functions, such as maintaining genomic information, apoptosis, autophagy, immune response, and regulation of tumor. In general, the role of this p53 protein is to trigger the production of other protein, which will act as a stopping signal for the cell division. In the case where the p53 is mutated, the cell division will not stop and will continue to divide. This will cause an abnormal growth of cells. This condition becomes really dangerous if the mutated cells are the ones that continue to divide, and this will further lead to tumor. This is how the p53 looks like. The blue one is the p53 and the purple one is the DNA. Now I will pass to Yip Teng to explain the key steps.
So now I'm going to explain 5 key steps involved in p53 transcriptional gene network. Let's talk about the first step in p53 transcriptional gene network, which is stress signal detection and p53 activation. At this stage, p53 is activated by stress signals such as DNA damage, oncogene activation, oxidative stress, or hypoxia. Then it becomes stabilized by preventing its degradation via MDM2. Second is the translocation to the nucleus. Stabilized p53 moves into the nucleus from cytoplasm, where it can act as a transcription factor and bind to specific DNA sequences in the nucleus. Third is the DNA binding and target gene activation. At this stage, p53 binds to specific DNA sequences and activates genes responsible for different cellular processes such as p21 in cell cycle arrest, GADD45 and p53R2 in DNA repair, or PAX, PUMA, and NOXA in apoptosis. Fourth is decision between repair and apoptosis. Depending on the severity of the damage, p53 either halts the cell cycle and initiates repair or triggers apoptosis if the damage is irreparable. Finally, its regulation and feedback loops. p53 levels are regulated by a negative feedback loop via MDM2, which ensures that p53 activity returns to normal once the damage is resolved. However, if p53 fails to trigger apoptosis in severely damaged cells, it can lead to tumor formation, which is a key factor in cancer development. In short, p53 controls whether a cell repairs itself or dies when there's damage. It helps prevent tumor formation by making sure severely damaged cells don't survive. If p53 fails, damaged cells can keep growing, leading to cancer. Moving on, Becky will explain more about the consequences of malfunctions.
The mutation of the p53 gene leads to many consequences. First is DNA binding dysfunction. The p53 gene controls cell growth, but mutations occur in its DNA binding area. It can activate genes like p21 and BES. This leads to uncontrolled cell division and cancer. Next, p21 activation failure. Normally, p53 activated the p21 gene to stop damaged cells from dividing. When this fails, cells keep replicating even with DNA damage, leading to more mutation and a higher risk of cancer. p53 gene's mutation also leads to defects in the apoptotic pathway. Normally, p53 helps trigger apoptosis, a process that removes damaged cells. But when genes like BES, PUMA, or NOXA malfunction, these damaged cells survive, leading to more mutations and cancer, such as lung cancer. Besides, we also got MDM2 overexpression. It means too much of these proteins, which binds to p53 and break it down. Without p53 to stop tumor growth, cells grow uncontrollably, leading to cancer. p53 helps fix DNA by controlling repair genes like GADD45 and SPC. If p53 is disturbed, DNA repair fails, leading to more mutation that can cause tumors. TP53 mutations disturb normal function of p53. This leads to common cancers like lung and breast cancer. It also causes Lee-Famandi syndrome, a rare inherited condition, which increases early cancer risks due to a faulty TP53 gene. Adeline will continue with the solution of these mutations.
Hello, I'm Adeline. So I'm going to talk about the solutions that you could go for once you have cancer caused by the non-functional p55 gene. So number one is PRP inhibitor-based chemotherapy. So this PRP inhibitor blocks certain enzymes for the cancer cell repair, so it actually causes them to undergo apoptosis. Number two is targeted therapy, which is by taking drugs containing inhibitors depending on the cause. For example, drugs with CDK inhibitors helps to activate p21 gene, and drugs with MDM2 inhibitors combining with DNA damaging drugs cause reversible cell cycle arrest. And lastly, drugs with histone deacetylase inhibitors activate the GADD45 without the help of any functional p53. Lastly, you could go for biomarker testing, where they will test for specific biomarkers so that you can look for other effective cancer treatments.